the abdomen that we are going to study today, we call them as liver, which is like largest organ that we have in our body. So as I am saying, this is a gland, that means it has a capacity of secretions. That's why that thing makes in a gland. So whenever you are going to study the gland, we are going to break it down into smaller fractions of a gland. We call them as lobules. So whenever you are going to study any of these particular glands, we subdivide or break it down to the smaller unit. We call them as smallest functional unit, which is we are going to see, or which are we going to see in the microscope. So we call them as lobules. And lobules comes from the lobes. So it's a gland which goes to break down to the lobes, and lobes breaks down to the lobules, and these particular lobules we are going to study in microanatomy. So whenever we're going to study this particular microanatomy of the liver, we are going to study this lobules or smaller fractions of this glandular tissues. And this particular gland is capable of doing secretions, and capable of doing the digestive actions. It's capable of storage and capable of defense action, etc., etc. And this is important, or the important metabolic organ in the abdomen content what we have. Now this particular lobule of this particular liver have got different way to describe. So this particular lobules in the liver, we call this the classical look of the liver lobules, or classical, if you add an adjective, what ideal liver lobes or lobules should be looking like. It should be looking like hexagons. So it should have 6 arms like this. Now this particular hexagonal look of this particular liver lobes or lobules is visible or can be demonstrable in lower animals. For the humans, we do not have this classical lobule look. For the humans, it is more or less like a spreaded. I cannot properly identified as this hexagonal lobule look. So this particular classical hepatic lobules, that should be looking like hexagons, and they are present next to each other. Like this one is, again they have another one, then we have this on the top of there as well. They are mingled with each other. So this particular classical lobule look is visible or can be demonstrable in low animals, lower primitive animals, that like rats or guinea pigs. When we pick up those particular mammals, we classically study this particular liver studies. But for us, for the humans, we do not have this classical pattern. Now this particular orientation of this particular hexagon pattern is contributed, or is formed by the presence of number of the connective tissues. So if you have more connective tissues in the livers, we get this particular structures, and this particular structure remains if you have more connective tissues, which is present in low animals. For us, we do not have that much of connective tissues. We have plenty of cells, or the liver cells. So that's why this particular orientations is not readily demonstrable in case of human bodies. But as we study this particular lower animals, we just try to gauge or try to figure out a classical hepatic lobules, which ideally should be looking like hexagons. All right. Now this particular hexagons has some features. So the central of this particular hexagon is pierced by a vein. We call them a central vein. So this particular thing, which is in a center position, which will be having a empty part, we call them a CV or central vein. Now on the margins where you seeing this particular hexagon meets, we have this particular union points. So this particular thing we call them as angles of the hexagons. So on these angles of the hexagons, they are 5 hexagonal angles or 6 hexagonal angles are there. Within this particular angles, 2 or 3 of them will be having kind of like a placement of vascular structures. So in these angles, we call these angles is present with some kind of vascular structures, which is present 3 of them like this. So we call this collections of these hexagonal arrangements of this particular vascular structure, we call them as portal triad. We call them as PT or portal triads. And this one we call them as CV, central veins. So these two orientations are there. Not all the angles should have this particular portal triad. Just like out of 5 or 6, we may have like 2 or 3 depending on these particular patterns of this particular hexagonal arrangements of the hepatic lobules. So two basic things that we are going to observe here. One is this central vein, that is the hallmark to identify the hexagons, or which is the broken down into the liver tissues or liver glands into lobes, into lobules. In each lobule should be looking like a classical hexagonal things which have 6 arms like that. The central of it will be pierced by a presence of a vein. We call them as CV. We call them as central vein. And these angles, we have these 4, 5, 6 angles are there. Within these angles, not all of them, but 2 or 3 of them will be placed with some kind of vascular structures. So these vascular structure collectively we call them as portal veins, PV, portal triads. Now out of these particular portal triads, there are 3 structures are demonstrable. It is a kind of 3 things. One is a branch of hepatic artery, HA. So the second thing we are going to see, a branch of portal vein and a branch of bile canaliculi. So 3 things are placed on this portal triad. That's why 3 things we call them as triad. We call them as portal triad. They are because of this particular taking the name of the portal vein branch of it. We take all this as portal triad, which is consisting of 3 structures of them. If you just like magnify, zoom them out, 3 of these structures are there, tube-like structures, which is a branch of hepatic artery, a branch of portal vein, and a branch of bile canaliculi. These are the orientations that we are going to see. So these are the basic thing which is present in it. Apart from this, particular liver cells are placed with this particular cells which is present in the liver. So these cells in the liver which is present, we call them as hepatocytes. So these particular hepatocytes, they are placed and placed rest of the places. And this particular hexagonal arrangement that you are seeing here, this particular hexagon things which you are taken over there, the shape of this is contributed by the presence of connective tissues. That means to give the shape of this connective tissue, collagen fibers, reticulin fibers. These are the fibers which are giving the shape of this or architectures of these things. Now, this particular hepatocytes which is present in these particular regions. How these particular hepatocytes are there? These hepatocytes are present in kind of like layers laid from this particular portal triad to this particular central vein. So like these directions they are placed like this. So they are arranged like in a radial fashion, radiating from this particular central veins to this particular portal triad or these hexagonal angles, right? So each of them, they have presentations like this. Now, between these particular layers or between these particular rows of the hepatocytes, we find a tiny space in between. So this gap which is present, which I am marking between these particular layers or rows of these hepatocytes, which is present in between this particular 2 adjacent rows of hepatocytes, we call them as space. And this space which is present in here, we call them as sinusoidal space. So we have a space which is existing between the 2 layers or 2 rows of these particular radiating manners of these particular hepatocytes. Between this tiny space, we call them as sinusoidal space. Now, around this particular portal triad, we have this space as well. So this particular space around this particular portal triad, between this particular hepatocytes and this particular portal triad content, we call them as space of Mal. So these are the 2 spaces which is present in here. We call them as space of Mal and sinusoidal space. Now, coming to here in this functional aspects of the liver. Now, as a gross structure in the liver, we see that it has got very rich blood supply, right? So it is very richly supplied with this particular blood to it. So there are 2 sources of blood which is coming to the liver. One source is coming from the intestines. So from the intestine it is coming as PV, portal vein. So all the nutrient rich content which is coming through here is coming on the portal vein, and portal vein is formed by the union of splenic vein and supramesenteric vein. We know that. So this is rich in nutrients. They are reaching to the liver, right? Other thing which to supply the oxygen, other things which is nutrients, everything, we need some kind of blood supply so that this hepatocytes and all this content remains live. That means we need a blood supply which is rich in oxygen. That means we have a blood supply coming from an artery. We call them as hepatic artery. So is a branch of abdominal aorta should be descending out and sending a branch to this particular content and supplying to it. So there are another source which is coming here is hepatic artery. So from these two sources, bloods are coming to this particular liver. One is from the hepatic artery and other is from the branch of portal vein. Agreed? Clear? So two sources feeding this particular liver. One is bringing up this particular nutrient rich content from this particular stomach, from this particular intestine. Other is bringing this oxygen rich content. Where they are emptying this particular blood? Portal triad. So this particular blood is emptied into this particular hepatic lobules, into this particular angles of these hepatocytes. We call them as portal triads. So we have 2-3 spaces there where this particular blood are coming from two sources, coming from the branches of the hepatic artery and branches for the portal vein. Now as this branch are coming, these blood are pouring through this particular areas of the sinusoidal space. Clear? And from this sinusoidal space it is traveling and reaching to this particular central vein. From the central vein it is going to merge with the central vein of the adjacent veins. So all these veins are joining together and eventually emptying into the vein which is bigger vein. What bigger vein we have in abdomen, which is draining the blood to the heart, we call them as inferior vena cava. So from this particular central vein, it is eventually draining to the hepatic veins, and hepatic veins eventually draining to IVC. Clear? So how this particular blood is flowing to this particular liver, between this particular liver cells, through this, maintaining with this hepatic architecture of hexagonal arrangements. Clear? Again, to start with, we have hepatic liver, which is for the studying purpose we are breaking it down to lobes. Then we break down the lobes into smaller lobes, we call them as lobules. And how do the classical thing of the lobules should be looking like? It should be a hexagonal pattern. We call them as classical liver lobules. That means that is the micro unit or functional unit of liver. Now, if you just like to see this, how this particular hepatic lobule should be looking like, it should be pierced at the central part with this hole, we call them as central vein, and it should have the 6 arms like this, which is formed by these reticular fibers or the collagen fibers, making this particular shape looking like this. At these angles of this particular hexagonal arrangements, we have this particular point, we call them as portal triad. So not all the angles should have this particular portal triad. The angles which they have, this particular collections of 3 units, branch of hepatic artery, branch of portal vein, and branch of the biliary canaliculi, then we call them as that is portal triad, right? So not all the 6 of them will be having, 2 or 3 maybe, or 1 may be having this particular angle. Now, for example, in this segment, you have 2 of them. In this segment, maybe there are 1 only which is isolatedly present. The third one may be having 3. So it is present in a manner which do not have a particular pattern that will be there, depending on like variations are there, depending on, so like usually we see the 2 to 3 angles which is fitted with this portal triads. And what is portal triad? Which is these 3 tubes or tubules that is present in it. Now, what are the functions of this particular element? We are going to understand. Then this particular liver parenchyma or liver, this particular lobules are placed with these particular cells. We call them as hepatocells or hepatocytes. Now, how these hepatocytes are arranged? Hepatocytes are arranged in rows, radiating manner, just like sun rays, they are radiating. So if we consider this one is the sun and the rays are going on the radiating manner and radiating towards this particular angles of the hexagons, and this particular rows, in between the rows we have a space. That space we call them as sinusoidal space. Now this particular sinusoidal space which is present between this particular adjacent rows of the hepatocytes. Likewise, between this particular portal triad or around that particular region, we have another kind of space. We call them a space of Mal. Right? There are separate other spaces there, space of dice, etc. are there. But these are the two things that is like to begin with is good with us. Now, between this particular cellular architecture of these hepatocytes, the functional part, how it functions, two of them we are seeing. As this particular liver is highly enriched with the blood supply, it receives the blood supply from two sources. One is the nutrient-rich sources, other is the oxygen-rich sources. That means it is pouring the blood from two different sources, and this particular blood is coming from the portal triad region and draining towards the portal vein regions. So it is coming from the portal triad to these particular regions. Now, you understand this particular area so far, and this particular central vein joins with this particular bigger veins, eventually makes this particular drains to this particular hepatic veins, and from the hepatic veins it drains to the inferior vena cava. So what kind of blood will be draining through this particular central vein? Oxygenated rich or deoxygenated rich? Deoxygenated. What kind of blood will be flowing through these particular portal regions? Oxygen rich. Clear? Oxygen nutrient rich. Clear? Because nutrients are also coming. Clear? So nutrients content will be more when you take the foods. After we are taking the foods, absorption is going on. So that time it is high in nutrient content. Later time it will be just like more and oxygen content, less nutrient contents. Clear? And as these particular nutrients are poured through these hepatocytes, hepatocytes are picking up these particular important elements that is need to be stored, keeping it for a while and utilizing as this metabolic process is going on, right? Depending on the hormonal stimulation, environmental stimulation, etc., etc. Now, so this region about this particular is more oxygen rich regions. Is the central part or the marginal part? Marginal parts. So if we have a deprivations of oxygen, that will be reaching to this central part or central canal. So if you have an oxygen damage region which is happening, is usually affects this particular region which is around the central veins. This region which is relatively have more oxygen content, and as the oxygen content is going on, this particular from this particular hexagonal angles to this particular central part, we lose the oxygen content. So if you have by any chance any damage or hepatic damage out of ischemic hepatic damage which suffers more in central region than that of peripheral region. Get my logic what I am saying? See, if the oxygen content by any means is less, the hepatocytes is going to suffer or hepatic lobules is going to suffer, right? So that particular content out of this particular hepatic lobules, the central part is worse sufferer than that of marginal part or peripheral part. Reason, the margins are more fed with oxygen than that of central part, as because oxygen flows from this particular region and slowly taken up this particular hepatocyte. So less oxygen is coming to the hepatocytes which is close to the central vein or central vein region. So this content of this oxygen distributions or this flow distributions that we have to keep in mind. Second thing, we are haven't talked with this particular element, we call them as biliary canaliculi. Now this particular flow of the blood which is coming from the portal vein and this particular hepatic arteries, they are to this direction towards this central vein. But this particular bile which is secreted from this particular metabolitions of hepatocytes are flowing to the opposite direction. So this bile flow is from the hepatocyte to this particular portal triad, because bile is going to this particular hepatic biliary canaliculi. Eventually they are joining together next to each other, making this particular hepatic bile content and eventually going to this particular central vein and eventually going to store to the gallbladder system, right? So these directions of the blood and directions of the bile is opposite in this particular hepatic cells. Get my logic what I am saying? Clear or not? So the directions of the bile flow and the directions of the blood flow is the reverse in case of this particular hepatocyte. Are we clear of this part or not? If you have any confusions, clear this, because later on it will be becoming complicated. So bile flow is from the central vein towards this portal triad, that is secreted from the hepatocytes. these hepatocytes which is present in this particular region, they are doing metabolism, and the metabolic products of these hepatocytes are secreted within this particular biliary small canaliculi, eventually going through this particular tracts and going through this tract, draining to the portal triad region and draining to the portal triad, small portal biliary canaliculi to the bigger portal canaliculi, eventually to this particular right and left hepatic bile duct, which eventually goes to the gallbladder, and from the gallbladder it drains out the cystic duct, through this cystic duct drains to this common bile duct, etc. Right. So this is how this particular bile flow and this blood flow differs in case of the liver system. Right. So having said that, this is the architecture of this particular biliary system or the liver cells which you are going to identify. So whenever you are going to identify this particular liver system, we are going to identify these things as hexagonal arrangement of the lobules, which is having a central canal. We call them as portal, we call them as central vein, which is having kind of like angles of this particular hexagons is fitted with this portal triad consisting of these three, and this radiating manner of the hepatocytes which is present in here, and between these hepatocytes we have this particular cells which is present in here. We have a space, we call them as sinusoidal space, which is flowing through the sinusoidal space, blood is flowing through and going in. So this is how this particular whole network of these liver cells is going on. Right, clear? Now we can divide this particular liver cells, we call them as classical hexagonal pattern. We can call this as portal triad as well, or portal lobules or acinar lobules. So if I just project it through this particular pictures, it will be clear now to understand what we are trying to describe out here.
So the bile flow is also via the sinusoidal space?
Bile flow is not through the sinusoidal space. Next to this particular, opposite to the sinusoidal space, we have this biliary space which runs through this particular, actually it is lying on the gutter system. So if you just look close, the sinusoidal to it, under to it we have a gutter, just like paracolic gutters, we have seen that. We have a gutter system which is runs below this particular hepatic level, one level down, and from that part it is excreted through this part and eventually going to. So it's not on the hepatosinusoids. Hepatosinusoid is for the flowing of the blood. This bile flow is to the opposite directions and takes the space which is opposite or next to this particular, under to this particular space, which is opposite side, but we call them as connective tissue, which is giving the structural support to this particular hexagonal arrangements, and classic hepatic lobules, which is hexagonal in shape. How this particular hepatic lobules are placed? We have a central, which is central vein, and from the central vein we have a radiating cords which is known as the hepatocytes, which is cells arranged in radially and moving towards this particular angles of this particular hexagons. And if this particular angles of the hexagon is fitted with this particular like veins or structures, we call them as portal triad, which is fitted with three structure: branch of portal vein and branch of hepatic artery and thus biliary ducts, interbiliary lobules, right? And this particular space between this particular like these hepatocytes, we call them as hepatic liver sinusoids. So this is the basic architecture of the things that we have agreed to. And liver in gross, it is a kind of like a modified exocrine gland, and this particular things is receiving the blood sample. Hepatocytes is the cell which is present in the liver cells and that has a particular pattern. Now blood flow to the liver is coming from two sources, as we have talked about: portal vein and hepatic arteries. Run through the hepatic sinusoids to central veins and to the bigger veins to the eventually to the hepatic veins to this inferior vena cava. This is how his blood is flowing through. So this is how this particular thing should be classically looking like. The hepatic sinusoids which look like the hexagonal structures, and angles of it, one or two maybe fitted up with this particular collections of this particular portal vein, hepatic artery and this bile canaliculi. Then we call them as portal triad. So this particular portal triad should be zoomed like looking like this. So we have a branch of portal vein, we should have a branch of hepatic artery and the bile canaliculi like this. So they are present like this, and the central vein over there. So the blood is coming from these angles and flowing through these hepatosinusoids, which is the space between here, which you call them as here, and running between and reaching to this particular part, which is central vein. This is how it is going and draining eventually. So this region, which is predominantly oxygen rich region, as it moves to the central vein region, it is less oxygen rich region. So if you have an oxygen flow problem, we have this particular, this hepatocyte suffers more than that of this vein. So based on this, how these particular hepatocytes are placed close to this particular portal triad, we can divide this particular area into 3 zones: zone 1, zone 2, zone 3. That means this one, zone 1, which is close to the portal triad. Zone 3 is most away from the portal triad, and intermediate zone, which is zone 2. So this zone 1 is oxygen rich region, zone 3 is less oxygen, so deprived of. So oxygen related damage, if you have any, then this particular zone 1 or this region is suffers more. So this particular region is having this particular hepatocyte loss. We see that in the patients more if you have any hepatic insult. Clear? Now, this is what we are talking here, is this particular arrangement of this particular hepatocytes. So this particular cords of hepatic cells, they are present like this. So we have the 2 rows like this, and between this we have this space. We call them as sinusoidal space, which is through the blood flows through from the peripheral to the central part, right? Now this particular sinusoids may have a modified kind of cells which is present in here, which is a modified endothelial cells, which is, we call them as Kupffer cells. These Kupffer cells are defense cells. They are modified endothelial cells which is lining these sinusoidal regions. So these modified endothelial cells, we call them as Kupffer cells. They are basically present in the sinusoidal space. They are modified endothelial cells. They are defense cells. That means modified monocytes, they are taking this particular defense actions or policemen which are present in this region. Now this is how this particular flow of the blood which is going, and the reverse flow of the bile, this should be looking like. So this particular flow of the blood is going from this particular portal triad region to the central vein, as we have mentioned out here, from this particular hepatic arteries to this particular portal vein. They are draining towards this particular central vein. The thing is that we have this particular thing which is bile flow, which is going on the opposite direction from this central vein region to this particular portal triad. Now this particular portal triad over here, the space which we call them as periportal space of Mal. So space of Mal around this particular portal region, they occupy there, and this particular smaller bile ductules joins with the bigger ductules and forms this eventually the hepatic ductules, right and left hepatic ducts or hepatic bile ducts, and right and left hepatic bile ducts unite to form common bile duct or common hepatic bile duct, which drains to the this gallbladder eventually, and this comes out from the gallbladder as cystic duct, and eventually they join with together with this common hepatic bile duct. So this is how this particular architecture there. So this space we call them a sinusoidal space, through which the blood flows here. Next to that, between this particular thing which is lying at the lower level or a level down, we have this particular bile flow tract. So this is separated from this particular sinusoidal space. This is 2 different space. So they are present in a different tier or different zone or different levels in terms of depth of this particular hepatocyte. They are not mixed together. If they are getting mixed together, we have a problem. So this particular bile is releasing into this particular blood system, and eventually they are mixing and flowing through the blood, and eventually getting this particular disease. We call this as jaundice. So this particular overflow or overspillage of the bile can happen. So this gutter, as I am saying, is small gutter. So what happens if gutter overfills? If you have flood filling up the gutters, what happens? It floods this area. So if you have overflow of bile, so by any chance this particular liver cells are secreting too much of biles, and which is overflowing this. So what happens? It should be lying at the level down. That means it should be a level down to the sinusoids. If it is lying on the level down over here, now if by any chance it is overflowing, বেশি flow হয়ে গেলে, যেমন river overflow হয়ে গেলে কি হয়? Flood হয়ে যায়. So same thing happens over here. Then this flood water enters to the localities. So these things also happens if you have overflow of bile, it enters to the sinusoidal space, and these things gets mixed up. So bile is directly entering to this particular blood system and flowing through the blood, and then we have this disease, we call them as jaundice. Getting this concept out here? So this thing, gutter things spills over, creates this particular problem. So you maybe have the excess bile flow or may have obstructions out here. So if you have a blockage here, for example, if you have a stone, for example, small biliary canicula stone occupied here, bile cannot flow here. What is going to happen? Going to spill, going to overflow. If it overflow, what is going to happen? It's going to flood the sinusoidal space. If it is flood the sinusoidal space, it is mixed up with the blood, and blood gets mixed up or contaminated with the bile. Now that blood is released into where? Portal vein. That portal vein drains to hepatic vein. That hepatic vein drains to IVC. That IVC drains to liver. The atrium. That atrium blood goes to ventricle, lungs, systemic circulation all over the body. Then we have this yellowish discoloration of the skin. So biliary pigment is all over the bodies. So this is how these things are spilling over, right? It could be various region, could be anything, right? So this is how this is going on. So this is the physiology of the bile flow or bile overflow or bile obstruction, etc., etc. So you have to understand this particular concept, then it makes easy. So if you understand this, how these things are going on with the two different systems, it's easy to understand this particular physiology and anatomy. So coming again together. So this particular element, we are going to make these things together over here. So this simple concept we are talking so far, right? So we have this particular hexagonal patterns, lobules, having three components over here: central vein, sinusoids or hepatocytes, and portal triad. And in between the hepatocytes, we have the sinusoids. So this part we have talked. Now we have learned this particular element looks like this. Now make it little bit complex out here. So between here, you see that we can divide this particular component over these regions, or divide this particular hepatic lobules. This is individual hepatic, we call them as lobules or sinusoids, classical sinusoidal hepatic lobules. Now you see that we have this particular central vein here, 1, 2, 3, three of them. If we join with the imaginary line, we get a triangular space. So this triangular space also consists of some space in the liver. Similar to that, between these two hepatocytes, so this one and that one, we get another space which is present in here. So based on these particular collections, we can get some kind of another kind of like a space which is present in the livers. We call this as a classical liver lobule that we have already discussed. Second thing we are going to talk about portal lobule. So it's triangular space. So if you join this particular space which is present in between this particular hepatic central veins joining here, we get this particular triangular regions, and holding in between the central part is a portal triad. So this particular region with this particular hepatic lobules, we call them as portal lobules. And again, this hepatic acini. So between this particular two corresponding central veins side by side, if you join this particular space, if you find the long axis and this particular horizontal axis joining together, we find this particular space here between these two adjacent hepatic central lobules. So we find this particular space spanning over here. We call them as this space as liver acini. So this particular liver acini can be divided into three domains, 1, 2, 3, that we have discussed based on how this particular thing is getting the oxygen. More oxygen, least oxygen, intermediate oxygen. That's why we can classify that. This particular hepatic acini, we count this whenever we are going to judge this amount of or depth of the liver insult that is happening in case of any kind of liver injuries. Maybe just like oxygen deprived injuries, or maybe any kind of injuries which is bringing this damage to the hepatocytes, etc., etc. For that reason, we are going to talk about this. So we call them as periportal zone, zone 1, then we have the zone 2 intermediate zone, which is like in between, and this zone 3, which is poor oxygen region, which is depending on what kind of what kind of pathogens or what kind of trouble the liver is getting. Based on that, we just like a divide on. In the pathology, they will be talking about this particular orientations. So they will be dealing with the diseases. So when they are going to deal with the diseases, they are going to talk about the classical lobules, they are going to talk about the portal lobules, and they are going to talk about the hepatic acini. And then they will be starting from here and building these things. So I am just introducing this part. They will be starting from here in the pathology classes, and they will be talking about the liver or liver related disease based on this particular acini pattern, or this particular portal triangle or portal lobules, or this classic hepatic lobules. So three different diseases they will be talking, and giving example. They are going to talk about the liver pathologies, right? So this architecture of the liver that we have to know. For us, we are emphasizing on classic liver lobules. That's what we are going to see. And this particular hepatocytes, how this particular bile flows from the hepatocytes, that we have to know. Going from the bile, which is secreted from the hepatocytes, going into the canaliculi, which is a lower level gutter, which drains to the bigger gutter, which is known as intrahepatic ductules, then interlobular, and even eventually to hepatic ducts, right and left, then to common hepatic duct to the gallbladder. This is how this flow of the bile is going on to this particular hepatocyte. So whenever you're going to see this particular hepatocytes microscopically, it should be looking like this. Again, this is a zoomed out picture. That means it's a kind of near 100x zoom, but we are not going to see this much of zoom. We are going to see that 10x zoom. So this particular thing, we are going to see the central lobules first, like this, and based on the central lobules, if you see that, then we're going to find this particular portal triad, which should be present somewhere here, or angles like this. So these regions you should be having this portal triad, which present like this, right? This is another region where we're going to see this portal triad like this. So these particular regions is looking like this, and in between this we have this particular hepatocytes. So you have to imagine through how these particular things, which will be looking like cords of hepatocytes, which is radiating manner they are going on. So we have two of these slides which are present in two of the specimens which are placed on the microscopes. Go through them, try to identify the central lobule, which is actually focused in the central part of the central lobules, and try to go for these margins where you are going to see the portal triads. So this is what we are going to identify, the central vein and portal triads on the margins of it. So not all the hexagonal margins you are going to see this portal triad, couple of them will be having